the online platforms are really helping us in ensuring that although our students are offline and the faculty who are offline are able to nurture the knowledge of the current status and current discoveries happening across the world. And it's an great pleasure for me to also share that based on my interaction with some of the industrialists in various areas of in particular in Hyderabad, I just want to share to all the participants also. Please understand that we are going to get the COVID-19 vaccine shortly, and as Modi promised by August 15th, it is going to come, because I even sent a message across that being a pharmacologist, I also want to be a volunteer for the clinical trial. However, I did not get the consent from the company. So hopefully if they have a space, they may even ask me to participate. And I also want to share that they will be complete by this month end, and it has started across India. And I think maybe it may take 15 minutes, 15 days, and maybe we may, if the results are positive, and I think we may get it, we may have the release date on August 15th itself. And hopefully with the National Institute of Virology keeping in progress, and Bharat Biotech already in full swing, right? And the clinical trials going on. Let us hope that we all come back to the normalcy, and maybe from September we all, or maybe later month, we all can have the vaccine and remove this pandemic from the world. And I think this is the time where India can prove themselves that we are already a pioneer in vaccines, and this will ensure that all of us will feel proud once this vaccine is discovered. So with a short note of this, I wish all the participants here that please remove the panic situation or the tension we have whenever we cough or whenever the temperature shoots up. So you need to take care for the next month end, and definitely we can ensure that we are going to get the vaccine. And this is an authentic information. It's not general information, and there are various controversies that you cannot prepare a vaccine overnight. All these things are there. Despite that, right, they have, the government has taken complete care to ensure that they are done with full protocols and at a faster pace. So this is a personal my message to all the participants here, and I wish this program all the success. And thank you so much, Dr. Praveen, for spending your valuable time. And I think we are very happy to have you with us. And I wish whenever you come to India, please ensure that you visit our place. And I would like to also acknowledge the thanks to the HOD of the Viper, Dr. Vanita, Bhavani, the convener, and all the team for successfully running the programs despite being Hyderabad having the highest percentages of corona positive cases, right? Thank you so much and wish you all the best. I hand it over to Bhavani or Sandhya or Sun, whoever it is. Thank you.
Thank you, sir. Now I request Ms. Sandhya Reddy to give a brief introduction about today's speaker, Dr. Pravin Shukla.
Thank you, ma'am. Good morning, one and all. Myself, Sandhya, Assistant Professor, Department of Pharmaceutics, Viper. It's my privilege to introduce Dr. Pravin Shukla. Dr. Pravin Shukla is an experienced pharmaceutical professional with a unique combination of preclinical clinical sciences and clinical pharmacology, with over 10 plus years of progressive experience in preclinical translational research, clinical pharmacology, and application of therapeutics in direct patient care. Sir has completed his diploma in pharmacy from AICT Mumbai. He completed his BS Pharmacy from RTM Nagpur University, MS Pharmacology and Toxicology from NIPER Chandigarh, PhD from School of Pharmacy, North Dakota State University. Sir has done his PDF in Stanford University School of Medicine, Stanford. Recently completed Doctor of Pharmacy from UCSF School of Pharmacy, San Francisco. Dr. Pravin Shukla has 50 publications in various peer-reviewed journals. Sir is continuing his services as editorial board member of Journal of Clinical and Experimental Pharmacology, Journal of Natural Science, Biology and Medicine. Sir is associated with California Society of Health System Pharmacists, American Society of Health System Pharmacists, American Pharmacists Association, Academy of Students Pharmacists. Sir was a finalist of UCSF 2-minute business competition. Sir has awarded with various scholarships, UCSF Walgreens Community Scholarship, UCSF Pharmacy Alumni Scholarship. Sir was awarded with NDSU Graduate School Research Award for Outstanding Research and Teaching Contribution. Young Investigator Award by American Physiological Society for Experimental Biology and Medicine. James T. Carlson Graduate Research Award. He was the first merit in Nagpur University in BS Pharmacy for all academic sessions. IPA awarded gold medal for academic in BS Pharmacy. Sir has published a book, Human Pluripotent Stem Cell Derived Cardiomyocytes from Target Assessment to Translational Biomarkers. Presently, Dr. Pravin Shukla is working as clinical pharmacologist to Vertex Pharmaceuticals Translational Medicine, San Francisco. On behalf of all participants, I welcome you, sir.
Thank you, Sandhya. Sir.
Thank you.
Yes, sir, please continue the session. Dr. Praveen.
Thank you, Ramesh. Thank you, Vanita, for nice introduction. And I'm really excited to see how we go about COVID-19 vaccine. And again, for providing that update. Just a quick thing. Everybody can call me Praveen. I think that is the best way to address me. I'm still a student learning. So with that, I will going to share. I believe we are ready to start the presentation now.
Yes, sir, please.
Yeah, we are. Okay. Okay, so as everybody have already heard about my background, I just wanted to give a little bit a different take to my background because see most of your audience is still in an early stage of their career. They are doing Bachelor of Pharmacy and Master's program. So this slide is not necessarily to show that how good thing I have done in my career, but just to show that every small thing you do basically build up, and eventually in long run all those things come together, which you launch a really good and satisfying career. So as already been mentioned, I did bachelor's from Nagpur University, and from NIPER I did my master's in pharmacology, and from NDSU I did my PhD in cardiovascular pharmacology. And from there I was at a conference giving a talk, and director of cardiovascular medicine from Stanford was there, and we were able to connect, and that's how I got recruited at Stanford to do a postdoctoral fellowship in cardiovascular electrophysiology. And more recently I have taken a transition from preclinical to more into clinical research, where I apply knowledge of therapeutics in tailoring drug therapy at bedside. And for that, I have done a doctor of pharmacy at UCSF School of Pharmacy. UCSF stands for University of California, San Francisco. So as you can see, it's been a long journey, and I can feel that being a first year B.Pharm student and or second year B.Pharm student, sometimes it might be little challenging to see how things will pan out down the line, maybe couple of years. So this slide just I want to show you to just exemplify one path how things will materialize one day. So stay there, stay strong, and I hope you are all. I hope all of you do really well in your didactics and eventually in your research career. So with that, I will pivot or switch to my today's presentation. Before getting into presentation, disclaimer is this is the work I have done between 2012 and 2017. So it is kind of a little older work of mine, which is more preclinical or translational research. For last, since 2017, I've been involved more in clinical trials, specifically in pediatric bone marrow transplant. And as Vanita reached out to me about this presentation, and we had a discussion which topic to pick, whether we should go towards bone marrow transplant route and see how we can apply principle of pharmacokinetic and pharmacodynamic to optimize therapy in little babies who are undergoing transplant. We decided that at this stage, perhaps discussing something like pluripotent derived cardiomyocyte and its application, and it's a broader application in disease modeling and drug screening might be much more helpful to the audience, which as I understand is predominantly B.Pharm and M.Pharm students. So so that's the reasoning behind us choosing this topic. So just to give you an overview what I'm going to talk about in next 1 hour or so. So the way I would like to start is to give an introduction that I already given to you. In terms of human pluripotent stem cell cardiomyocyte, which is a very mouthful, it's a short form is iPSC, which is induced pluripotent stem cell, or it could be HESC, which is a human embryonic stem cell. And I will tell you the difference in between and why, what is the property of each of those. So we're gonna see how this whole research field came about in 2005, 2007. What was the motivation? And why we need or why we should care about stem cells, whether it is induced pluripotent or human embryonic. What these cells offered, what unique advantage these cells offer, what promise these cells hold that we should care about or spend time doing research on it. After that, I will go into a little bit of technicality, like what are challenges using these cells, why we are still not able to use, which could range from developing small organs, transplant, or doing drug discovery, screening at a large scale, and eventually treating some of the blood disorder or the liquid disorders. And after that, I will move on to, despite all the limitation, how we can work with these cells. So, okay, you can make these cells, how you can characterize these cells, what are the different ways you can characterize. And there I will bring my expertise as an electrophysiologist, that how I can record ion channel current in a tiny cell and see how ion channel current changes as we change the physiology, or we perturb the ion channel dynamic by using a therapeutic molecule, be it experimental or already in therapy. And then I will touch base a little bit about what are the challenges, why we are still not going forward with this technology in a manner that we should or we ought to be. So there are some of the challenges that I'm going to share. So this will be the groundwork. This will provide the foundational understanding of what is the current landscape and what are some of the limitations. And then I will going to switch a little bit and share details of a project that that is yet not published, but where I was able to, I and some of my colleagues at Stanford could develop a really nice model by using an array of assay where we use cardiomyocyte derived from iPSC to model arrhythmia or arithmogenicity induced by hypokalemia and also the antiarrhythmic drug. So that's in nutshell what I'm going to talk about. So as I mentioned, what is the current landscape? So how this whole field started? What is a pluripotent stem cell? What does pluripotency means or multipotency means? It literally means in a very common sense of way is that a cell can be differentiated into different type of cells. So is something like a mother cell. A stem cell can respond to different biochemical cues or microenvironment and can differentiate to become bone cell, a blood cell. It can become skin cell and a hair cell and pancreas and liver and kidney. You name it. So this cell has within its DNA to become different kind of cells. So for example, neuron or heart cell, those are somatic cells. So somatic cells are the cells which are terminally differentiated. Those cells during our lifetime do not differentiate into any appreciable extent. Whereas the liver, if you liver has still some regenerative capacity, if you as you can see in case of liver transplant. So that's what pluripotency means, the ability of cells to differentiate into different kind of somatic cells. Now, there are two different kind of pluripotent stem cell. One is when the fertilization happened in womb, you can take those embryo out and you can isolate those cells, and those cells are called human embryonic stem cell. The word embryonic refers to the fact that they came out of a fertilized embryo. The second is as you can imagine, there are many ethical issues in terms of getting those cells. So this is an example. This picture just shows like how the blastocyte stage embryo looks like, and this was first isolated by Jamie Thompson in 1998. The problem with that is big ethical issue, especially in United States. Is are we willing to use embryo to generate stem cell? And there have been multiple attempts, at least early in 2000, to ban the funding for any research which involved embryonic stem cell. Now, so is there any other way we circumvent the ethical dilemma of using human embryo, destroy them and use the stem cell? So in 2006, in Shinya Yamanaka, and he was working at UCSF where I did my PharmD. He basically figured out a very elegant and simple way, first in mouse and then in human, where you can literally take your skin cells or any somatic cells which is accessible. You take those somatic cells and then you put 4 transcription factor in those somatic cells. And those 4 transcription factor were enough to trick a fully developed somatic cell to travel back in time and acquire pluripotent. So this cell become in some way similar to human embryonic stem cells. So you take a somatic cell, you introduce certain 4 transcription factor, and that lead to the induction of pluripotency. And once the cells become pluripotent, we call it induced pluripotent cell. The word induced signifies that we are inducing them to become, versus embryonic, which signifies that it came out of an embryo. Now, both cells share, both cell types share a very common properties. That one, they are pluripotent, so you can differentiate theoretically. You can differentiate them into different kind of somatic cells, and I'll share how we can do how we can make a cardiomyocyte. You can also make and pancreatic cells and even hepatocytes. So despite their origin, the one unique factor between one difference between embryonic stem cell and induced pluripotent is that for induced pluripotent cell, you can make these cells from each individual human being. So imagine a scenario. I mean, this is again a hypothetical scenario. Is that a patient had some disease that can be corrected by making X. So literally, I mean, that's what the ideal that just will be the ideal situation where you have that patient come in, take his skin cells, and you transform his adult skin cell, which are somatic cells, into pluripotent stem cell, and then you trick those cells biochemically to become your cell of interest, and then you put those cells back into the same patient, and that way the patient will not even reject those cells because those cells came out of the same patient. So that's really unique about human-induced pluripotent stem cell, and you can make these cells anytime in a patient's life. Whereas embryonic stem cell can be made only when the embryo is in the womb, right? So that's the, and of course on top of that there are ethical issues. So that's the one unique thing about that. But despite this major difference, both cells have some unique properties, that they are immortal in a sense, like they will you can keep propagating them in a culture media, and they retain pluripotency. I mean, you can have a cell today and you can maintain those cells for perhaps several months or a year, and you come back and those cells will still be dividing and growing. They have very stable proliferative rate. As I mentioned, they proliferate very well, and you can culture them, differentiate efficiently, and they become a renewable source of differentiated cells. So you can imagine if ever we figure out a way to make an organ out of a iPSC cells, we will have a limitless amount of iPSC cells that we can create, and hence the limitless amount of somatic cell of our interest that we can make. And if we can figure out bioengineering, we can use those cells to make different organoids. So that has a potential. Again, I'm using the word potential to rectify the lack of of healthy organs for several patients who is waiting on a waiting list for several maladies, starting from liver cirrhosis to heart diseases. So this just shows that there are some biochemical marker, and given the level of our audience, I will not go into the molecular biology, but these are the marker which just tells you is that your adult somatic cell have become pluripotent stem cell. They have crossed that barrier. As I mentioned, once you make either embryonic or iPSC, you technically can differentiate these cells to become either cardiomyocyte, endothelial cells, smooth muscle cells, neurons, red blood cell, adipocyte. I have personally made myself cardiomyocyte and neurons, so I have very good experience. Like, these cells are fairly easy, at least in vitro assay system, to differentiate into cardiomyocyte and neuron. So what we want to do with it? Okay, you got your iPSC cell. What are the different things you can do? As I mentioned, these cells can differentiate into several adult somatic cell type. You can also use these cells which may have a genetic disease, and you can correct that genetic disease and then differentiate in a healthy cell type and put it back into patient. Or you can also use these cells for other purposes like disease modeling and stem cell disease modeling and drug discovery and toxicity screening. So this is the idealistic future we would like to be, where every becomes or her own donor. So patient has a disease with a clear genetic cause. You can take the disease cells, do the genetic engineering, make healthy cell, and infuse back into the patient, theoretically curing the patient one and done. Or a patient doesn't have a clear monogenic disorder that is amenable to correction. You can take iPSC cell and you can basically develop an organ, and you can transplant that organ back to the patient in case if you have an organ-directed disease like kidney disease or liver disease or heart disease. So this is our idealistic picture, and I'm hoping that in our lifetime we get to that point. But what is stopping us? Why? What are the steps, or what are the questions that we need to answer before we realize this dream of ours? So these are some of the questions that I can think of. And as I mentioned, like, people and different research group have been trying very actively, but these are by and large major questions that we need to answer. The first question is, can we produce a GMP, which is a good manufacturing practice and good clinical practice grade iPSC in large quantities with a very high yield? And the second would be is, can we differentiate this iPSC into the cell of our choice? Can I make a lot of cardiomyocyte? Can I make a lot of neurons? And the second question is, once you make those neurons, you have make those neurons in a Petri dish or perhaps in a big flask. Now the question is, how close those cells in terms of molecular signature, functional phenotype, and some of the structural component of those cells are close to an adult somatic cells. Because if somebody has a heart disease or liver, you want to make an organ which is capable of performing the same function like a 20-year-old or 30-year-old or 50-year-old person. So how we can characterize and ascertain that these cells are as good as your adult somatic cells? And if they are not, what are the ways we can make them become as mature as adult somatic cell, both in terms of molecular signature, genetic signature, and the functional phenotype? Now, since I'm going to talk more about cardiomyocyte, which is my interest, how well these cells, so that's the in terms of maturity and application in organ transplant. But in terms of if you want to model these, if you want to use these cells in terms of modeling, then the question is how well these cells, when I say these cells, means somatic cells that you derive from iPSC, like cardiomyocyte, how closely these cells recapitulate the human disease. So for example, if I want to develop a model of arrhythmia and I put a genetic mutation in sodium channel, can I recapitulate the disease phenotype, both in terms of its genetic signature, biochemical signature, as well as a functional phenotype signature? And if it can, how well that model will respond to drug that we already know can rectify that disease. That's basically the model validation. So these are some of the questions, and I hope to answer at least partially some of these questions and share a project where we were able we were able to demonstrate the utility of iPSC-derived cardiomyocyte in recapitulating the human disease and drug response. So the first thing is, when this system came about or came online, our ability to produce these cells were really low. So and I'm specifically talking about cardiac cells. So you got your human embryonic stem cell and you are trying to make beating heart cells. So back in 2001, when after Jamie Thompson came up with that method, you would get only 1 to 5% of yield. So for example, so you can easily guess, if you theoretically, if you do 100 cells, you might get 1 to 5 cardiomyocyte out of those cells. And that was very time consuming, unpredictable, and it has a very low yield. I mean, you could not get enough cells to even characterize or develop a model, let alone make a human organoid. Now, as happens in science and in life, with enough dedication and work, things get better if you apply effort in right direction. And as you can see, these are some publications that I won't go in detail, but the idea here was as the time passed and people had a better understanding of differentiation biology, they came up with a better and better method. And they basically, this is the one which is shown in green that came from Palchik group. And this is the method came online when I started my postdoc in 2012. And at that time, we could consistently get more than 90% of pure cardiomyocyte. And eventually, by the time I finished my postdoc, we could get as high as 95-98% of cardiomyocyte out of the induced pluripotent stem cell. So now at least we have the input material that we can make patient or humans or a particular person specific induced pluripotent stem cell, and then we can biochemically trick them to become a beating cardiomyocyte. So now you have access to human cardiomyocyte that we never had in a way that we never had. Because before that, all you can do is you might get a small strip of a heart muscle tissue from a cardiologist who is doing some kind of surgery or ablation, or he's cutting off cutting off your atrial pouch. So then you basically you basically digest that tissue and get some cardiomyocyte, and you hope that you can record the current. But again, you can imagine those cells will die off in maximum 6 to 8 hours, and that's pretty much it. You never had access to human cardiomyocyte physiology as we now have with the iPSC technology. So if you just take a moment to absorb this breakthrough, you will imagine the potential this technology hold. So this is a book chapter that I wrote with my advisor, and this just shows you a particular way we go about making these cells and characterizing these cells and making sure that what we are making is what it is actually. So I already shared you can have a human embryonic or induced pluripotent. You basically differentiate them and you do the key marker. You make sure that those cells have key markers, so that's kind of quality control. Now you know that they have all the markers, and then you basically figure out a biochemical cocktail that you can incubate your stem cells, and then the cell will basically differentiate into any somatic type of your choice. In our case, we were interested in making cardiomyocyte. And when you have a heart cells, you can imagine what we're interested in. The one key thing about heart cell is basically the membrane electric excitability of the membrane, and which comes from the fact that they have a very dense high density of ion channel and calcium handling. So those assays basically come about based upon what kind of underlying biology cardiomyocyte have. So you will gonna do as electrophysiology, you're gonna do some immunostaining. You can check how well these cells are handling calcium, because calcium is important for contraction. You just record action potential, which shows that membrane can rhythmically basically let the current flow through the membrane, which is action potential. So those are some of the assays you can do. Then you can do flow cytometry, protein expression, and gene expression, where you basically figure out whether these cells are expressing all the right genes that a cardiomyocyte should have. So we are basically using a very huge battery of tests which basically apply to characterize this cardiomyocyte. And here the idea is to confirm that the cardiomyocyte you're getting is actually a cardiomyocyte in every shape and form, whether it's a gene or whether it's a biochemistry or whether it's an actual function. So I believe the video is not working, but that's why I wanted to show you a Petri dish where the heart cells are actually contracting. So this is a sheath of cardiomyocyte, and if I'm pretty sure the video isn't working, but if I could show you this video, you can imagine this sheath is synchronously beating, and this is a very amazing sight to see. And I still remember the first time I differentiated cells and saw the cell beating inside the microscope. I mean that was the that was the most amazing thing I've seen as a fellow. So the second, as I mentioned, is you can take these cells and you can stain them. So this color, each color means a particular marker. So green color means actinin, and then red color means troponin. So troponin is very specific to heart cells, and you can, as you might know, when patient undergo myocardial infarction, we usually check for their troponin level, which signifies that the heart cells are dying and the troponin is being spilled in the blood. So the red is showing troponin, and then you just overlay both the images, and you can see that these are cardiomyocyte, and although not as mature as it should be, but these are cardiomyocyte. And you can see a very nice sarcomeric structure, and the blue here shows the nucleus stains. So now, if you have a heart cell, as an electrophysiologist, I need to know, is this cell beating? And the second, can I record electrical activity in that cell? That is the action potential. And I believe I just included this slide because I believe BPharm student can easily correlate this with one of the chapter of Goodman-Gilman or Lang pharmacology, where you see that the action potential generates because of a rhythmic synchronous opening and closing of a bunch of ion channel which respond to membrane depolarization and repolarization. So just to quickly walk through this phase 0 is a membrane depolarization. It is principally mediated by inward sodium current. And the phase 1 is just mainly because of outward potassium current, a very slight one. And then phase 2 and 3 are again by the outward potassium current and then inward calcium current, and which lead to membrane depolarization and calcium influx. That's called membrane excitation contraction coupling. And then you have phase 3 or Herc current, then you have phase 4. So the reason I'm showing you is that if my cardiomyocyte that we just made can reproduce this kind of action potential, that gives me confidence that, okay, these cells are cardiomyocyte, because this is the key signature of a cardiomyocyte, that it will produce action potential like that. But how well how well our cardiomyocyte express these ion channel? So this slide is a little complicated. It is much more deeper in research. But what it's trying to show you is that how the gene expression of these cells is happening in your pluripotent stem cells. So the question is like, are they as good as the adult heart cells or not? So this slide just it shows you like to what extent you are, to what percent or to what extent you are recapitulating the adult cardiomyocyte ion channel current and as well as expression. So just to orient you, if you look at the top blue row, what you see here is the current density. So that's the column, this column. Then you see gene expression, that's this column. And then you see the one in the red or light pinkish color, that is your pluripotent stem cell. And the blue is referred to the fetal cardiomyocyte, neonatal, and adult cardiomyocyte. So let's just take an example of sodium channel. So if you look at sodium channel, both current and gene expression, it is quite comparable. So the red is basically our cardiomyocyte that we have just made from either induced pluripotent stem cell or human embryonic. And what you see is that it pretty much closely overlap in terms of current density and also in terms of gene expression. So this gives me some confidence that, okay, these cells, even though they are just 30-day-old or 40-day-old, their current density and gene expression for sodium channel is as good as adult cardiomyocyte. Excuse me. And so this is how to read this chart. And I'll just gonna highlight some of the key differences in these channels. So these just show that our cells, the pluripotent stem cell cardiomyocyte, they have a very lower L-type calcium channel. And then they have a little bit of a T-type calcium channel, and we don't want T-type because this reflects more of a immature or fetal phenotype. And these cells tend to have something called HCN, which is a funny current. So this current is important for which gives a heart, which gives SA node and AV node. Lot of these ion channel current in your cardiomyocyte, because what will happen is your heart cell will beat, but in actual adult heart, only SA node, AV node send out the signals, and in response to those, the adult cell contract. But what happens with our cardiomyocyte that we make, they have very high level of HCN, and because of the presence of this HCN, you have a lot of large funny current, and that funny current make them beat. So imagine if all of your heart cells are beating on their own and not listening to SA node and AV node firing rate, something called overriding phenomena, then you are in trouble. So you want a ventricular myocyte which is mature enough to do so that it doesn't contract on itself, but it will contract if you depolarize by putting electrode, or when these cells are listening to the SA node or AV node, the impulse validated by these nodes, and that's what we want. So this presence of a large HCN current, you can see the pink color, and as you can see the blue color. The blue color is going down as you start from early fetal, late fetal, neonatal, and adult. You see that adult cardiomyocytes literally have no HCN current, but our cells tend to have a good amount of HCN current even after 30 days, and those that make these cells to beat by themselves. And it has a very great implication, given that if you inject this cardiomyocyte in a heart, or if your pluripotent stem cell becomes heart cell inside your body, this may or may not cause arrhythmogenic effect in your body. So these are some of the what should I say, immature phenotype of our cell as opposed to the cardiac cell. And there are some more that I can point, but the idea I'm giving you out of this slide is our cells are good, they look good, they have a good phenotype, they produce action potential, but they still lack significant similarity with the adult cardiomyocyte that we would like to see. Okay, how we can characterize. So this slide just simply shows that these are the cells that our group at Stanford made, and I was an electrophysiologist which characterized. So this is just for reference, just to show you that how the characterization look like. So you have marker of pluripotency, so you establish that these cells are pluripotent, and then you have a cardiac marker that I just showed you, and then you have a very high fidelity electrophysiology assay where you record current in each cell, and each cell will give off different pattern of action potential trace. And if it is this pattern, you can call it nodal like. If it is this pattern, it's atrial like, and this it is a ventricular like. And as you can see, we have something called Lqt, and Lqt phenotype is nothing but a long QT phenotype. So there are some patients who have some mutation in their cardiac ion channel gene, which make their repolarization longer than what you and I would have. And those are called long QT phenotype. So this patient has this gene, and we could easily, so these cells that we made out of those patients can actually produce the same phenotype that we see clinically on an ECG in an intact human being. So that's really a big leap in terms of our ability to capture EFI signature of a cardiac disease from a somatic cell to pluripotent stem cell and back to somatic cells. In addition, these cells are pretty robust. You can use a normal iPSC cardiomyocyte and you use some gene editing technique, and you can put a gene. So for example, this mutation M1766L, and this just to give an example, if you have a mutation which is known to cause a particular form of action potential perturbation, that is a prolonged action potential by increasing late sodium channel, you can actually insert this mutation by using one of the very advanced gene editing technique like CRISPR, and you can actually recapitulate the same phenotype. So it means these cells have the underlying physiology of a cardiomyocyte, in as much as if you tweak a one ion channel which governs the action potential, you can see what will be the perturbation, what will be the perturbation of mutating that ion channel in terms of the electrophysiological phenotype, which is again, as I mentioned, it's very, very thrilling to see. So this is a very raw recording that I recorded, and you can see these are the real numbers. It's a real time. You have a seconds and you have millivolt. So this is unedited. So you didn't put any ion, you did not introduce any mutation, and then you did the introduce mutation, and as you can see, just by visibly glancing, is that you have a longer action potential duration. And what this waveform is basically one heart cell beating. The another thing that I mentioned is we also need to know, like, these cells could respond to known drug, because we know those drugs pharmacology, and we want to validate that this model will respond as it should. So here is the example when you take these cells and you pace them. So when you pacing means you're basically forcing them to beat slower. So whenever a heart cell beats slower, its action potential goes longer. And as you can see, when you beat it lower, you have a longer action potential. But when you increase the pacing, so 1 hertz would be 60 times per minute, and 0.25 hertz would be 1/4. And you can see you can actually abrogate this prolonged action potential. So you can pace these cells. And then you can use, since we are all pharmacists or pharmacologists, you can use some kind of a pharmacology. You can hit this cells with ranolazine or maxilitine. These are a putative late sodium channel current blocker, and they will block this late sodium, and you can see. So the blue is the disease phenotype after adding the mutation. Oh, sorry. And ranolazine is when you add, you can actually rectify the prolonged action potential. That's really fascinating to see that these model this cardiomyocyte respond to changing the ion channel function, either by a natural mutation from a patient or by artificially injecting a mutation via CRISPR or TALEN technology, and how these model then respond to drug. So this gives us a lot of confidence that these cells, however immature in terms of ion channel expression vis-a-vis adult somatic cell, is still recapitulate the ion channel dysfunction. So this is the paper that one of my friend Arun Sharma and I was, it was a big team we did, where we wanted to show that can human-induced pluripotent stem cell recapitulate the cardiotoxicity induced by tyrosine kinase inhibitor. So this is a usual workflow that I have been showing you, that you take a somatic cell, you reprogram them by adding the 4 transcription factor, and then you differentiate into either cardiomyocyte, endothelial cell, and cardiac fibroblasts. And once you have these cells, then you basically go on to test any class of drug. And in this case, we did the tyrosine kinase inhibitor because these drugs are known to have a very strong cardiotox signal. So this I was responsible to do the electrophysiological experiment, and what you see is that each action potential, and that this line show you that when it was recorded. So this is a continuous recording, 0 minute, after 3 minutes, after 5 minutes, and after 10 minutes. As you see, when we treat cell with the vehicle or 4 different tyrosine kinase inhibitor, you see that you don't see any change. But when you incubate these cells for at least 2 hours, you tend to see that nilotinib and vanditanib, these two tyrosine kinase inhibitor, which we know from clinical trial that can cause long QT, we can recapitulate the effect in the cells. And again, this is really fascinating that how we can recapitulate the human cardiac biology and the pharmacology in terms of both therapeutic as well as the toxicology. This just gives you overview that what we have done so far. So as you can see, there are several diseases that different groups around the world have modeled using induced pluripotent stem cell. So this is a very big list, and anybody who is interested can go to PubMed and they can pull out the paper. The idea here is the same that I showed you right now. You recruit patient, you characterize pluripotent stem cell, and you make somatic cell. Then you basically validate the model by either gene editing or by rectifying with the drug. And this has been a very exciting work in our field. Now, any new technology comes with some challenges inherent to it. Going from B Pharm first year to 4th year is also equally challenging. I mean, at least when I was doing my bachelor's, every year I go from 1 year, first year to 2nd year, I feel, oh gosh, this is so challenging. So in that spirit, coming to challenges. What are the challenges? The biggest challenge that, at least with respect to cardiomyocyte, is that these cells are not functionally mature. So if I have a 50-year-old person, I make cardiomyocyte from his skin cells, those cardiomyocyte will not reflect the maturity of an adult heart cell. And that is something that we need to, as a scientific community, to figure out how to mature these heart cells. Because you cannot use those cells until those cells are equally mature as the adult somatic heart cells. So this is how adult heart somatic cell look like. Oh, sorry, adult heart cell look like. So basically, this is the staining of a sarcomere, actin, and cardiac troponin staining. And you can see this cell is really straight, multinucleated. It is very well stratified, and there's a striation, and this looks perfect because that's how the heart cell should be. But what we are getting, so if you go and differentiate 1 billion cells, this is what you end up getting. You still get the sarcomeric structure, but the structure doesn't have any pattern. It still has a nucleus. The cell is live. The cell has gene expression. Cell produces action potential, but cell is not as functionally matured or structurally matured as an adult cardiac myocyte should be. So going into how they look like, how we should take a cardiac myocyte produced by induced pluripotent stem cell. One way to think about it is like these cells in terms of maturity are more fetal-like versus adult-like. So we want them to be adult-like, but currently the kind of cells we are making are much more fetal-like versus adult-like, right? And this slide is really dense, and but the idea here is like they have a structural, electrophysiological, and calcium handling features which shows immaturity when you compare with them with the other adult cells. So the next challenge would be is what kind of biochemical, mechanical stimuli we should be feeding these cardiomyocytes, which forces them to become mature, closer to adult-like and not fetal-like. So that is, I would say, that is the holy grail. And I think there are several groups which are making a lot of progress in terms of cultivating these cells on top of a string and then making them string to stretch in a different direction, and hoping that that stretching we will going to realign the sarcomere and make those cells mature. But again, we need to a technology where we can scale up these cells to billions and billions of cells without losing the quality, and this all needs to be done under GMP and GCP. Okay, so I think so. After giving you the foundation, and I know some of the stuff might be little complicated to explain over slides, but I just wanted to give you a flavor that how this technology came about and what it means, and how we can play with it and what we can do with it. Now, I'm gonna share a project that I was leading. Is can we use this cardiomyocyte to model hypokalemia induced arithmeogenicity? So just a little background why we care about hypokalemia and why we care about hypokalemia induced arithmeogenicity. So if you see in the clinic, especially in an older patient who have some kind of pre-existing heart disease, hypokalemia becomes a really big problem. Patient who have a kidney disease or who are on dialysis, both hypo and hyperkalemia becomes a very big problem. And why it matters to heart? The reason it matters to heart is because potassium plays a very important role in modulating the membrane excitability, and hence how the cardiac myocyte will respond to SA and AV-induced signals. And you can imagine if a heart has some kind of lesion and you are exposing that heart into hypokalemia, then it will be like an insult which might exacerbate the baseline heart condition. So that's why we thought, can we use our iPSC cardiomyocyte and hit them with the hypokalemic environment and see what kind of a phenotype we can expect. And on top of that, we wanted to see is what happens, how the heart cell will respond to antiarrhythmic drug or any, as a matter of fact, any drug which can perturb cardiac ion channel physiology under normal potassium and low potassium level. Because low and high potassium is basically modulating your membrane excitability. And then on top, you have a drug like terfenadine, or you have cisapride, or you have, you name it, any QT prolonging drug like moxifloxacin. And you want to see like how it is going to affect phenotype. So this list is not necessarily for you to read, but I'm just trying to show you, like, there are there are several drugs which are known to cause prolonged QT or TDP. TDP is torsade points. So a patient who has a really prolonged QT can go into really bad arrhythmia, and that arrhythmia is called TDP. So what it tells me is that there is no pattern. I mean, there are so many drugs, their chemical structure is different, their pharmacological class is different, their target is different, but they tend to have some risk of TDP. So what is the pattern? What are the factors which will help us predict that which drug will have which drug has TDP risk or not, or if there's any model that we can come up with to assess the risk of these drugs. So again, bringing the same point is it is highly unpredictable and very rare to assess the drug-induced TDP. TDP is just, in your mind, you can take TDP as a really bad, life-threatening arrhythmia. The estimated incidence of this event, for example, with cisapride, which is well-known prokinetic drug and which causes long QT and TDP, is 1 in 111,000 prescription. So it's very difficult to model any study, be it preclinical or clinical, with enough power to detect a signal which is 1 in 111,000 prescription. That's very rare. So we wanted to see that is there something that which can help us predict. So one could be is that patient has a pre-existing cardiovascular disease or some undiagnosed lesion. What it means is either a patient has a heart disease or patient doesn't know he has some kind of ion channel mutation, but that mutation hasn't produced any, the mutation hasn't manifested in a form of a cardiac disease. So that would be subclinical disease. And the third would be a secondary reduction in repolarization reserve, which could be happen because of offending drug or electrolyte disturbance for hypokalemia. So what it means in simple term is it's basically a backup plan. So in human physiology, we have redundancy in terms of pathways. If your one pathway fails, body still has a second pathway. If that fails, there's a compensatory pathway. So same heart has a multiple ion channel which let heart cell repolarize after depolarization, and that's called repolarization reserve. So for example, you have a IKR current, you have IKS current, and the idea is even though you have one ion channel defective, the other ion channel can still try to cope that defect and let your heart beat in as much of a normal fashion as it can. But imagine if you have a one genetic lesion in your heart and then you add a drug, which is a second insult, or it could be hypokalemia plus drug plus a underlying disease, then everything comes together and that will lead to the development of a very, very bad arrhythmia, and that might lead to sudden cardiac death. So the idea was, can we model that? We cannot have a we cannot have a patient who will volunteer to model this kind of study. The only way you can do is you take the cardiomyocyte from a patient with a known disease, cardiomyocyte from a healthy patient, expose them to the hypokalemic environment, which is first insult, and then you add a drug which can perturb ion channel, which is second insult, and see how it changes the how it changes the cardiac action potential profile. Can you see some kind of perturbation happening or not? So that's what the basic idea or motivation behind this project. As I mentioned, we really don't know which factor will trigger hypokalemia, and this just shows you a big laundry list of the factor that can cause hypokalemia, that can lead to that will enhance risk of torsade points. And this list also goes on top of this list, which shows that, and you as a B.Pharm student know that each drug class has a different chemical structure, and chemical structure basically defines how or which drug target they're going to bind. So you can imagine you really can't get a pattern out of these drugs, that which drug or which chemical structure is predictive of causing TDP. And the same goes on to this factor. This is a very heterogeneous factor. You have an impaired hepatic clearance of drug, diuretic use, patient has a congenital long QT polymorphism, patient can have like a female gender. So there are so many factors. So this is the model we came up with. This is what we wanted to do. We wanted to have a cardiomyocyte which with a disease. It could be a cardiomyopathy, dilated or hypertrophic, or it could be long QT. Then we add a facilitator like hypokalemia. And then on top of that, we add a trigger. That could be inotropy, epinephrine, norepinephrine, and that will stimulate the heart. Or you can add some drug like anti-arrhythmic drug, which has some pro-arrhythmic potential, and see what kind of phenotype you get. This is a very high-level view of the experimental design, but what it is telling you basically is that we recruited patients at Stanford Hospital. We did the genetics, we means the whole team, did a genetic screening, and we found out 30 patients who had a long QT hypertrophic cardiomyopathy or diabetic cardiomyopathy disease phenotype. We got their somatic cell, made iPSC, characterized cardiomyocyte, and then use those cardiomyocyte and treat and expose them to hypokalemia with or without anti-arrhythmic drug. So this is a little technical slide, but what I would like you to imagine is this is a single heart beating. Excuse me. Just imagine this is a single heart beating, and you have put an electrode inside that single cell. And this cell is beating very happily. It is beating, as you can see, this blue line. It is very rhythmic. It is not skipping any beat. Its amplitude is fine. Everything is going hunky and dory. So this is the normal potassium that your and my heart cell is bathing right now. And then what we did is that we did the pacing. We stimulated the heart. By adding norepinephrine. As you all know, it hits the beta-1 receptor, causes heart to beat faster and faster. And as you can see, the red part, this is the same time scale. Now you can see more beats are together and the beats are getting smaller, right? And you can take this information and you can plot this data. Without going into what this plot means, just visualize if these points on the plots, if the closer those plot points are, that shows that your heart cells are responding in a nice way. The more this point goes away from this central line, that means your heart is responding in a very arrhythmic pattern. So what happens? So this is happening under normal potassium level in a healthy human heart when you record without norepinephrine or without or with norepinephrine or isoproterenol. Now think of the same thing if you lower the potassium level. So same cell, normal cell, no disease, nothing. And when you lower the potassium, you see what happens. And this is a very long recording. This is a 20-minute recording from a single cell which is beating. As you can see, the cell is beating very slowly. There is some arrhythmic pattern, so it is not a normal rhythm. Arrhythmia is loss of normal rhythm. So you don't have a normal rhythm. And when you stimulate with the isoproterenol or norepinephrine, you can see the heartbeat pattern is getting uniform. But as time passes, as you can see, the heart goes into a sort of a single cell arrhythmia. So it is beating very erratically, and here it is basically stopping completely and then going into a big burst of arrhythmia, stopping and then going into big burst of arrhythmia. And that is happening in a healthy human cardiomyocyte when you lower the potassium level and challenge that cell with the isoproterenol. How you can imagine in your mind is, imagine an athlete who is playing and who is doing training for a long time, and his potassium has gone down, and he is pushing his exercise endurance, and his heart rates keep going up and up. Imagine if that patient, if that young athlete has some kind of mutation or heart disease, that patient will gonna that athlete will drop dead, and that's called sudden cardiac death or SCD. And so I'm just giving you putative idea to use to put this result in perspective. Okay, this graph, as I showed you already, I told you like if these points are clustered together, your heart is beating rhythmically. The more spread out these beats are, it means the more your heart is getting into rhythm. So when I show you this plot, the last plot, you can see that all the heartbeats are all over the place, and that's the classical definition of arrhythmia. As I already mentioned, this just shows you the same data that how. Uh, so the black plot is show the low potassium. The blue shows when you initially add norepinephrine, the heart start beating very well, but slowly with time, the heart loses its rhythm, and you see that this red dot, it's going all over the places. And this is just a different way of showing the same data. But the point here is, if you lower the potassium and you raise your heart, there are good chances that your heart will respond in an arrhythmic fashion. And this is just the same data, just showing in a different way that what happens normal and when you have a hypo. And you can see these graphs, how neat and tidy these points are on the left side. And as you move on the right side, the chaos of the point increases, which is a harbinger of a arrhythmic pattern. Now, so this is all is happening at the electrical level. Electrical level means heart is beating and you're recording action potential. But heart also has calcium handling, and you know some of the calcium channel blocker, verapamil, deltaenzyme. These are the calcium. They block LTCC, and they are used for arrhythmia, right? So once your ion channel has open, once your sodium channel has open, membrane depolarizes. The second event that happen is the opening of calcium channel, and the predominant phenotype is L-type calcium channel. We call LTCC, and the opening of that channel causes the influx of calcium inside the cell, and that then calcium basically go bind to actin, myosin, and that lead to the actual contraction, and that's how the heart pushes out the blood, which we call as a stroke volume or cardiac output on a minute basis. So we wanted to see, like, beyond membrane excitability, what is happening to calcium handling. So here what you see is a membrane contraction. So membrane contraction is a function of calcium, and calcium entry is a function of membrane excitability. So what you see, so in this assay, each waveform is not action potential. This waveform is showing the contraction. So as you can see, 5.4, when we go from 5.4, which is normal, down to 2.5, you can see this contraction is becoming slower. And you are having the less number of contraction as opposed to the normal. And this is just basically the plotting of the same data. But if you can just visually go from top to bottom and look at the level of potassium that is in the chamber, and just by lowering potassium, you can also lower the contraction. Now, this is a little complicated slide and much more technical, but what I'm trying to show here, what this slide is showing, that one of my colleague at Stanford very painstakingly did, is that when you lower the potassium, it not only changes the membrane excitability, contraction, but it also alters the way your heart cell handles calcium. So what happens when your heart cell contracts each time, lot of calcium spills out from the sarcoplasmic reticulum, and that calcium bind and then to actin myosin, there's a contraction, and after that the calcium is taken back into sarcoplasmic reticulum, and that rhythmic cycle makes sure that your heart is in a rhythm. But what's happening with this is that when we low potassium, it compromises this rhythmic kinetic and lead to the elevation of diastolic calcium. So what it means is, in systole, when your heart is contracting, you want the intracellular cytoplasm to be flooded with the calcium. The more calcium, the more contraction, right? That's the inotropy, or the force of contraction. But the moment the contraction is done, you want all the calcium to just sequestered in a sarcoplasmic reticulum or taken out of the cell. because calcium is a very important signaling molecule, intercellular signaling molecule or second messenger. You don't want calcium to just lay around in the cytoplasm while cell is in relaxing mode or diastole. So if any signal which is causing your cell to increase diastolic calcium will gonna bring some trouble. So that's what hypokalemia does. Is the hypokalemia increases the level of calcium inside the cell when the cell is relaxing. We don't want that calcium to be there when cell is relaxing. We want that calcium to be inside, nicely tucked away in sarcoplasmic reticulum and release only when the cell undergo contraction or the systole. Now, so far what we have seen is the functional assay. Action potential, contractility, calcium handling. Now, is this all is happening at the gene expression level? You can imagine if somebody is having hypokalemia or a patient who have a congestive heart failure, who are on diuretic therapy for a long time, what is happening at the gene level? Does hypokalemia changes the gene expression level, which ultimately lead to the phenotype? So this is the RT-PCR data. So this is just what each graph shows is the magnitude of ion channel expression. And the black is the normal, and the red is the blue, and the red shows the hypokalemia of different intensity. So I would like you to just eyeball each ion channel and just look at the black bar and then look at the blue and the red bar. And you can see across the board, there's a down regulation of pretty much every ion channel which is involved in membrane, which is involved in a rhythmic excitation contraction coupling of a heart of a cardiomyocyte actually. So as you see, IK1, it is important for resting membrane potential, and then you have ITO, which is also important for maintaining the membrane potential. Then you have Hur current. I think this is the human ether go related. It's a very complicated name, but the Hur channel, it's the IKR current. This is a very famous current that ion channel that you might know from your textbook. Because drug like terfenadine, cisapride, dofetilide, ibutylide, these drug block this channel, and that's how they prolong the action potential. And in other words, they also prolong effective refractive period. So these channel, you can see that there's a decrease in this ion channel level. That means there's a prolonged QT. And then there's the IKS, which is another repolarizing potassium channel. And again, this is the ryanodine receptor, SARCA, and L-type calcium channel. All of them are showing a pattern which can be construed as that there's a compensatory increase in SARCA expression to sequester hypokalemia-induced elevated intracellular calcium. So just to simplify, the message I'm driving home is not necessarily the technicality, but the idea is presence of hypokalemia perturb the calcium membrane excitability, calcium handling, both at a functional level. Functional means as in ion channel function changes, as well as the gene expression, which gene expression also changes. It means one, how the ion channel function, and two, how much of that ion channel is there also changes when the cell experiences hypokalemia. Now, if I had to put this all together, this is a very simplified model based upon this data, which tells you is that if you have a hypokalemia, it basically lowers all the potassium current, which is responsible for repolarization, which prolongs the action potential duration, causing early after depolarization. If I were you, I would not worry about the acronym, but the idea here is hypokalemia causing calcium overload and reducing potassium current. And that basically lead to arrhythmic trigger. And if somebody is interested, I can send some of the article for you to read. But the main message out of this slide is calcium overload and decreased potassium. That's lead to decreased repolarization through potassium ions and potassium ion channel that is causing the arrhythmia trigger. Now, one can easily deduce that what should be the next step. The next step should be, can we use now this model to see how the antiarrhythmic drug will respond if in a control and a long QT iPSC cardiomyocyte. So you can imagine if a patient who has some genetic defect causing long QT and a patient who is and patient who doesn't, and then you expose to both hypokalemia as well as a drug which has a potential to cause proarrhythmia. As you all know, all antiarrhythmic drug can cause all the antiarrhythmic drug inherently have a proarrhythmic potential. More so a drug like dofetilide, ibutylide, which is a very pure IKR blocker. So this was a really interesting experiment. We chose three antiarrhythmic drugs. One is dofetilide, this is amiodarone, this is flecainide. And amiodarone has the lowest risk of causing QTP. Flecainide has a moderate, and the highest risk has a dofetilide. I just finished my clinical training at Stanford Hospital, and during my cardiology rotation, I have seen 3 to 4 patients who went into Torsades de Pointes or experienced really bad arrhythmia when they were started on dofetilide. So dofetilide is a really has a high risk of causing QTP and TDP. And what you see here is a different concentration of drug, and each concentration is coded with a color line, and it shows that what is happening to action potential. So the black means the baseline in all three, and you see that when you add 300 nanomolar of dofetilide, you see the action potential becomes so long, and then there's a early after depolarization. That's basically is a signal for the arrhythmia might happen. With the amiodarone, even up to 3000 nanomolar, you do not see any sign of early sign of arrhythmic event. And with the flecainide, you really has to go. Flecainide has little more dose response relationship where at a concentration around 10,000 nanomolar, which is really supratherapeutic, where you tend to see EAD. Now, the cool experiment to do here is do the same thing in presence of hypokalemia and see how soon or at what concentration the cell starts showing the arrhythmic signature. So here you see the dose-dependent effect of flecainide on a long QT patient. So patient has some kind of gene defect. That's why its a QT interval is longer than what it will be for you and me. The therapeutic plasma concentration that we see in patient of flecainide is 300 to 2000 nanomole. So keep that in mind and correlate this concentration range with this concentration. So highest is 2000, right? So you see what's happening between 1000 and 3000. You can see between 1000 and 3000, there's really no extra beat happening in the action potential. But at 10,000, which is, I would say, 5-fold higher level of therapeutic concentration range, you see development of EAD, and that's happening in the long QT. So this is the baseline, and then you continually keep adding this higher concentration. The cell eventually becomes stopped. The cell stops beating. And we know that the cell is not dead because if you remove that drug and keep washing the cell, after 10 minutes, you regain the same action potential like here. So this just shows that how these cells are responding under normal potassium. Now, what happens if I lower the potassium? So let's lower the potassium to 3.5 and keep everything same. That means flecainide and long QT. So this is your just hypokalemia by itself causes the prolonged action potential. So patient already has long QT. Hypokalemia is making it become longer even more. And then you just add 30 nanomolar of flecainide, and you see 30 nanomolar is 10 times lower than the lower therapeutic range of this drug. So even so cells become so sensitive is that even if you add 10 times lower concentration, the cell start responding in a arrhythmic fashion. And here it is 100 nanomolar. 100 nanomolar is still 2 or 3 times lower than the therapeutic range. And what you see is the cell is literally undergoing in a kind of a ventricular tachyarrhythmia. So this model very beautifully captures is that just lowering the potassium level, you can lowering the potassium level unmask the proarrhythmic potential of a drug, which is actually a moderate risk drug, that is flecainide. And then So this is just putting this all together. So what did we learn from this? I mean, this seems very interesting. We can publish paper out of it, but what is the new thing that we learned? So we could show that this is the model which is predictive of a drug causing TDP. One is the patient has an underlying lesion. When I say lesion, means some kind of a damage in a heart cell. It could be structural damage. It could be ion channelopathy like long QT. And then there has to be a facilitator. The patient might be experiencing hypokalemia, acidosis. It could be something else as well. And on top of that, there's a trigger. Trigger is inotropy. It means heart is racing fast, or there's some kind of cardiac stress, or it could be drug, like in our case, flecainide or dofetilide. So these iPSC-derived cardiomyocytes very beautifully captured the model that we proposed, and we could demonstrate that these cells, albeit with all the limitation in terms of functional maturity and differential ion channel expression, which is much more akin to fetal versus adult that we would like. But despite all those caveats and despite all those limitations, the model is still respond in a very predictable way to known drugs and respond very well to hypokalemia and shows the complex interaction between these three factors to generate a arrhythmic model. So that's what we learn out of it. With that, I think it has gone longer than I hoped. If you have any other question, I'll be more than happy to take your questions.
Yeah. Thank you, Praveen.
Yeah. How much time do we have? I think it's how long it was.
It's okay.
Okay.
A stop sharing. Yeah.
Okay.
Yeah, I request Sandhya to ask the question. Sandhya, are you there? So we have a couple of questions, sir. Sandhya?
Yes, ma'am. Thank you, sir. Thank you very much. It was a very wonderful session and informative in depth knowledge of human pluripotent stem cells in terms of cardiomyocytes. And I hope this session will give a lot of insight to our participants in the view of in vitro and in silico molecular studies. So, sir, with your permission, I would like to present some of the questions raised by our participants. Can I go ahead?
Can you call me Praveen, please?
Okay, Praveen, sir.
No, just Praveen. Thank you.
Okay.
Sir, in our academics, we have the tradition of calling everyone as Sir, Ma'am. So that is the reason we are not coming out of that.
Okay. So then just call me Dr. Shukla.
Okay. Okay, sir. So can I raise questions on behalf of participants? Okay, sir. The first question was raised as like, what are some misconceptions of the stem cells in the molecular research?
Misconception as in in what context? That's the all question you have?
Yes, sir. Yes.
Well, since I don't have much information, so I'll just use my own judgment. Misconception in a sense is, I'll just use my experience, is that when you think of a stem cell and we see these news, news cycle, and we see some news, okay, now we can make pancreatic cell, we can make heart cell, we can make neuron. So suddenly in public imagination, the things comes, oh, okay, now we can make heart cells. So we should be able to cure heart disease, or we can make pancreatic cells. So we be able to cure type 1 diabetes. And India is a big, is almost diabetic capital of the world. But I would say the misconception would be it's a long road from making something in a petri dish and then doing the then doing the enough studies to make sure that that goes into humans safely. and has some efficacy and does not have any untoward effects. And where I live in California, here there are many clinics which claim that stem cell can make you look better, and they inject stem cells in eyelid and then God knows in which part of the body. And there have been some report that how those stem cells have become bone inside your eyelid. So I would say that those are some of the misconception I came across. But I would just leave with the thought is any new tool or technology should be taken with a big grain of salt, because every technology has its own arc of development, and this applies to everywhere. When I was in my master's program, buying a phone was very costly. You cannot make a call, and now we have a really cheap phone. So every technology had its own gestation period, and it is more so when you talk about medication in the field of medicine, where we are dealing with the life of human and trying to understand the human biology. So when the stakes are that high, the room for misconception will always be very high.
Okay, sir. The next question is regarding what is the difference between embryonic and umbilical stem cells?
In the major difference is in their name. So embryonic stem cell, you are catching as the embryo is forming at a blastocyte stage. You are pulling cells from there. In umbilical stem cell is basically you are catching it during during the birth. Both cells have a pluripotency. So when I say pluripotency means they have an innate ability to become somatic cell. It just the embryonic stem cell you take at a different stage, and the embryonic and the umbilical stem cell you take at a different stage. And I know increasingly people are banking umbilical stem cells when their baby is born, and the idea is like in the future down the line, if God forbid something happens, you can thaw those umbilical cells and do the same chemistry, the same biology, and make, I don't know, make some kind of organ, and so that you can save the baby's life. And the other thing with that is, at least in bone marrow transplant or when there's a liquid cancer, CML, AML, there people are using umbilical cord blood for transplant. So that's where human embryonic stem cell haven't gone there. So that will be another big difference there. Yeah.
Yes, the next question is how quickly we can develop this cardiac stem cells for treating neonatal emergency cardiac complications from umbilical stem cells.
Yeah, I mean that's the $1 trillion question actually. I would be very, very conservative and humble. I would say I don't know actually. I was part of the study funded by SUM. SUM is a California Institute of Regenerative Medicine, and we were trying to put these cells into the pig heart. So we will damage pig heart, and then we'll try to inject these cells. And the idea was that these cells will become heart cells, replace the damaged part, and the pig heart will beat better. So we had a limited success in that area. So I would defer this question to future scientists who are attending this seminar, and some of them will go on to figure out all these cool new discoveries.
Yes, thank you, sir. The next question is related to the pandemic situation and correlating with the cell therapy. The question is like, has cell-based therapy the research value for future treatment of COVID-19?
Can you repeat the question, please, for me?
Has cell therapy based the research value for future treatment of COVID-19? Will the cell therapy give a research value for future treatment in case of COVID-19?
Yeah. I know there's some, yeah, of course. I mean, it's a cell model. You can use cell model to infect with different viruses, bacteria, and just see what happened to the cell. And I know that there's some literature in this area where they have used iPSC cardiomyocyte and infected those with the coronavirus to see like what kind of damage those viruses doing to those cells. And the motivation, know what a different organ where COVID is causing damage, and at least in the kid, it might damage heart as well. So that is the one thing we can do. We also know that we can make lung epithelial cell can be generated from iPSC cells. So you can basically use those cells to put COVID-19 and see how it is affecting the lung epithelial cell, because it's primarily affecting lungs. To the extent we know, I mean, it is going to a different organ as well. So yeah, I would say to the extent any cell model will be used to investigate effect of any insult, to that extent, yes, iPSC derived somatic cell, be it lung or heart or neuron, can be a good model to test what COVID can do.
Okay, the next question is, can we make lung cells using pluripotent cells and use for cancer studies?
So that's a little out of my expertise, but I have, I'm not very fresh with that literature, but I just by knowing the field, I know that we can make a lung epithelial cell out of the iPSCs. It's just a matter of understanding the biochemistry. What specific chemical cocktail will trick the iPSC to become whatever you want them to. So, like for example, I have tricked them to become neuron or a cardiomyocyte, a fibroblast or endothelial cell. All you need to understand is what biochemical cocktail you should expose your iPSC for it to become the somatic cell of your choice.
Okay. Thank you, sir. It's very much clear regarding how we can develop the cell culture using pluripotent stem cells. Just one more question. Is how this human-induced pluripotent stem cells can be used for treating thalassemia or improve the quality of life of thalassemia patients?
Yeah. Actually, thalassemia is so everything that affects blood disorder, there's a better solution to that than iPSC. I mean, of course, you can get the iPSC, you make hematopoietic stem cell, you correct the gene defect, you infuse back. So recently, if you know the company named CRISPR Therapeutics, so this CRISPR Therapeutic is doing that trial. They take out the stem cell from bone marrow of the same patient and they gene correct the defective gene using CRISPR, and then basically they infuse back the gene corrected hematopoietic stem cell back to the patient, and that is much more efficient way of doing it right now. Because if you take iPSC and you make them hematopoietic stem cell, you still get into those same question: are they mature enough? You need to well characterize them. But if you take the bone marrow and get the hematopoietic stem cell, do the gene editing, put them back, you don't need to worry about immune rejection or any kind of heavy doses of immunosuppression that you need to give in case of a allograft, which happens in bone marrow transplant. So those are some of the advantage. But again, thalassemia is not my my primary expertise. But that's what I would say, and I would direct your the person who asked question to literature, I think, in this case.
Thank you, sir. Giving us a review regarding thalassemia, as till of now we don't have the treatment for thalassemia. So for in such cases, the participant has raised a question. Yes, you have given a good reference regarding the company who is developing and going through the clinical trial. The next question is that regarding the software, is there any specific software that can analyze volume, scarcity, and population of cells through the image of cell culture in Petri plate?
I wish we had something like that. it would have solved so many problems. I mean, you can do some kind of image analysis, AI, artificial intelligence, but the question is what answer you want. Do you want a quick, superficial answer, or do you want to really thoroughly characterize the cell? If, for example, if it's a neuron or cardiomyocyte, if I need to know whether this is a true heart cell, whether it can produce action potential, there's no way any imaging technique can look into the flask and tell me. All the imaging technique can tell you is, okay, what kind of shape, structure, staining, and all that kind. You need to have, so basically the assays are of a different, different value of the assay. And each assay will gonna give you a different level of information. If you want really deep information about the particular system, you really need to have a very high, what should I say, an assay which is designed to give you that answer. But if you want to just look at the cells and the health of cells, and yes, you can take a quick look over the microscope. But that will, how informative that will be. And again, the choice of assay will depend upon what question you are trying to ask. If I want to know if my heart cells have a long QT or to do patch clamp or need to put an electrode inside the cell, there is no way I can look at the cell and see it. So yeah, so it depends upon what question you want to get, what question you are trying to answer. That will should dictate the choice of technique.
I think it's regarding during growing the cell culture. While growing the cell culture.
Well, I'm not the cell biologist basically, but I can easily see what it means is like, you can look at the turbidity, how turbid or cloudy the media is, which can tell you how much the cell volume would be. But that is much of a manufacturing aspect of it. You are just doing manufacturing and you want to know that how much the flask is growing, and yeah, I think we can borrow some of the technique from microbiology for that, I guess. That's not my area of expertise, but I see the point in question here. Yeah.
So the next question, as we have seen some plasma therapy has been carried out for COVID patients. So the question is related to it. The effectiveness of mesenchymal stem cell and convalescent plasma therapy in COVID-19 patient. Which is better treatment therapy?
I really don't see a point of mesenchymal cell here. You have an active virus dividing. You want a passive immunity. So when you are giving plasma, what you're giving? You are giving passive immunity, right? It is just like giving the sera with a preformed antibody. Yes. So yeah, I mean, if it's up to me, I'm gonna give you the plasma with a well characterized antibody, neutralizing antibody, not just a binding. So binding and neutralizing antibody, really, and preferably in a high concentration in the sera. So yeah, of course, that's the number one. I really don't know how mesenchymal cell would help in an acute infection which lasts for a couple of weeks. Because anything which goes through stem cell takes longer time to. First of all, I don't know what is the purpose of giving mesenchymal cell, but assuming somehow this might help, the therapeutic modality will not fit the disease course, right? So disease course is what, 3 weeks, 4 weeks, right? And also the disease intensity. If somebody who is really sick with COVID, you need neutralizing antibody. Yeah. Unless you have a specific inhibitor which can stop replication of virus.
Thank you, sir. Related to cardiac study that you have presented in the graphs, so one of the participants raised the question that will isoproterenol stimulate only potassium ion channel?
Isoproterenol will stimulate beta 1 receptors. And the beta 1 receptor, the downstream to that is a different ion channel. Yeah.
Okay. So only ion channel will be having the isoproterenol will be giving more impact, will be having more action or the impact on the potassium channel. That's it, sir. Or whether there will be having any more ions like calcium, magnesium, even these ions are having functioning. I mean, these ions are functioning, helps in functioning of the cardiac muscles and the cardiac rhythmicity. So while studying the cardiac related rhythmicity.
Yep, I agree. Yep. It definitely, so downstream to the giving isoproterenol, downstream to that, it will going to affect the, because when your heart is beating and you are trying to increase the heart rate, the only way you can increase your heart rate is by decreasing the amount of time cell has spent in diastole, right? Like for example, if you are to run faster, the only way you run faster is by having a more number of steps taken. The more number of steps you take, the less time you have to relax, right? The less time you have to relax, the more tired you're gonna get, right? So same is the heart cell. The ion channel, the way ion channel works in the heart cell is by going through a cycle of repolarization and depolarization. And those membrane potential change basically dictates the closing and opening of channel, activation, deactivation, right? So if you put isoproterenol, definitely it will gonna slow down all the potassium channel, because the heart will spend less and less time in diastole and more time in systole. That's the only way you get you put more heart rate in the same time versus the normal, right? You increase number of heart rate in the same time. So that will going to increase the resting membrane potential, means it will become more depolarized, and your all the ion channel will not be coming back to their resting state again because of the cycling, right? So that's what I could think. But I know there are some specific effect of isoproterenol on potassium channel, and that's skipping my mind. But yeah, there's some data to show that what is specific channel and how it modulates those.
Thank you, sir. Just one more question to go. As we have conducted so many webinars till date, like it's a fifth day, but the questions are just coming as like there is no stop for the in the live. Also, we are getting so many questions from the participants. I think this is one of the best interactive session we have till date since 6 days. So, can I raise one more questions?
Yeah, sure. Go ahead.
Yes. Can we manage to use the stem cell? Can we manage to use the stem cells based tissue in clinical trials instead of human trials?
Instead of
human trials, can we manage to use stem cells based tissues in clinical trials? I mean, in the trial sessions instead of using humans as a volunteers.
Well, that's a very ambitious goal, first of all. And second, humans are made up of 9 different systems. As we know that there's multiple feedback and reflex systems, so all the system works together. But I understand why the question is being asked. So what is happening right now is we are trying to develop an organ system. So for example, liver. Can we make a liver in a small scaffold by putting stem cell? Can we make a lung model? So we can make an organ level model. So that's called clinical trial in a chip or clinical trial in a dish, right? So yeah, that's what they are trying to do, because they, so imagine like we in our evolutionary history, I don't know, however millions of years, as a human being in our evolutionary history, this is the first time ever we can even make a beating heart cells. It's just like 20 years. So I can imagine eventually we will be in a position where we can make at least one system from a stem cell and make it as close to physiology as possible. If you make a liver, you can do the metabolic study. You can use a new compound and see how it is metabolizing. Because right now what we do is we use a human hepatocyte culture. And then we get the metabolite and we do the MALDI-TOF and HPCL. When I was finishing my PhD, I interviewed at Wyss Institute in Harvard. And their Dong Inber, they were developing an organ in chip model. So they basically, for example, think of your lung. So lung has a smooth muscle cell, and then you have a very flat epithelial cell, right? So you can actually develop a scaffold where you incubate the interior with the epithelial cell, and then you on the top you put the smooth muscle cell and let them grow. So you are just trying to develop a model of your trachea or model of a bronchi, you know. So organ in a dish, organ in a chip is it will be a reality soon. But how soon? I'm not sure. Yeah. But we understand, we know the logic behind it. We understand the know-how. It's just the technology or our understanding of biochemistry and biology need to catch up with our ambition. And I'm very glad that your audience has that such a high ambition, because in their lifetime they should be able to do all those things. Not that I can't. I mean, I'm not that old, but imagine. Yeah.
Yes, very good, very useful information. Let's hope for possibilities of such organ in chip in the future.
Yep.
Yes, sir. One more question.
Yeah.
Yeah, otherwise, sir, can we communicate these questions in your mail ID, or otherwise it is getting late for you? Is it okay?
No, let's do one more question. Yeah, it's almost
Yeah,
midnight
it's
in
the last question, Sandhya.
Yeah.
Oh, okay, madam. There are any ways to overcome the immaturity of transplanted human pluripotent stem cells, cardiomyocytes, and also the lack of electrical integration, which may lead to arrhythmogenic risks in patients?
Yeah. So I think there are two questions in your one question. The first question is, do we have any way of forcing them, our fetal-like cardiomyocyte, to become mature? Can we force them? And the second is, if these cells are not very well integrated, that will lead to the generation of arrhythmia when you put in a patient, right? So I think the answer lies in the first part, because cardiomyocytes are really well connected. There's a connection in between so that the electrical impulse goes pretty fast. I know there are several groups which are trying to take the cardiomyocyte, make a small tissue ring, and then put that ring between two poles and move the pole mechanically. So that way you get a stretch like the way your heart is getting. Like if you look at the baby's heart, they're beating pretty fast. So you want to train those fetal-like cardiomyocyte by this kind of action. The other way is like you can have a electrical wire and you can coat your cardiomyocyte, and then you put a current inside that wire. Thereby you are pacing your heart cells. You are pacing at a 60 beat per minute, 80 beat per minute, or whatever beat per minute. The other way is you can have a cardiomyocyte and you can put some kind of a light activated ion channel in the heart cell, and then put those cells under the microscope with a particular light source, like UV light of a particular nanometer, particular wavelength, and then stimulate it with that. And the idea here is you are training your immature heart cell to beat at a particular chronically, and then see that cell become mature. But which sounds pretty good. But imagine we are doing it a very scale this up. You need to scale this up pretty high. So we know how to do it, but is the best way? I think that remains to be seen. And can we scale up? Can we scale up to GMP or GCP level, like good manufacturing, good clinical practices? That would be a big question, I would say. Yeah.
Thank you, sir.
Thank you, Sandhya.
Session. Thank you, madam. Over to you, ma'am.
Thank you.
Thank you, Praveen.
Yeah, no problem.
Presentation.
Yeah.
Actually, we have a flow of questions today. Like being in your busy schedule, you have taken some time for us to share your knowledge and expertise, research expertise. Thank you very much. And we are expecting your support in future also. We extend your support in future also. Thank you very much from all the participants.
Thank you, Vinita. Thank you, everyone.
Dear participants, thanks for your participation. Tomorrow we have session at 10 o'clock. Please join for the session. Thank you.